The overall goal of the following experiment is to demonstrate a single-molecule mechanical unfolding experiment on an RNA structure. In the first step, a flow chamber is constructed and mounted to the instrument. In the second step, micron-sized beads are mixed with the RNA sample of interest, and then the two different sizes of beads are loaded into the flow chamber. Finally, the flow chamber and the optical trap can be used to steer the beads into contact with one another to evaluate a variety of single-molecule interactions. Ultimately, this nanomanipulation technique can be used to study the folding of RNA at the secondary and tertiary structural levels.
This method can help answer key questions in the RNA folding field, such as how does an RNA fold and unfold, how does it transit between different conformations in response to an environmental change, and how does it interact with other molecules.
Today's experiment will be performed by my graduate student, Mr. William Stephenson.
To make a flow chamber, begin by drilling 6 2mm diameter holes into one No. 2 cover slip. Then use a laser engraver to cut 3 2mm wide slits into 2 pieces of double-sided polyimide tape. Attach one of the pieces of tape to the cover slip, making sure to align the holes in the tape with the holes in the cover slip. Attach the other piece of tape to a clean, undrilled cover slip. Remove the protective covering from both pieces of tape, and then place a glass-pulled micropipette and 2 bypass tubes between each of the channels on one of the cover slips. Next,